Okay, so I've talked, I'm almost done. Primarily the role of tracking transplanted cells, so I think, is image-guided delivery. So we use catheters and needles, and we make sure that the cells are injected at the right place. From a remote site, we can see when they're home. And then we have a number of other questions that are more difficult to answer. For instance, how long do stem cells survive? With these techniques you can tell. Do stem cells form teratomas? Do they keep dividing? And also, do stem cells differentiate? If you transplant a neural stem cell, does it become a dopaminergic neuron? And also when? Now I'll give one example of a technique that can answer some of these questions, which is bioluminescent imaging, that uses the luciferase gene from the firefly. You've all seen them in June. You can transfect mammalian cells. When you inject a substrate, you can do that IP, luciferin, then light is produced. We can put a reporter gene in two ways. It's called a constitutive expression. That means the cells always express that, but only when they're alive. So this tells us the cell survival. We can quantify the light flux. So this is the number of days after transplantation, and you see that these cells do fairly well. Now it's amazing when you do this kind of transplantation studies, what we found out, how standard drug regimens of immunosuppressant often do not work in rodents, even though they're routinely given, and people don't look at the survival over time, they just assume that this is the best way. Now when we place our reporter gene under a promoter, that only gets turned on if the cells decides to differentiate, then you can see a time course. In this case, the promoter is opalin, which is a kind of a marker for oligodendrocytes that myelinate. And you can see it takes a while. These are undifferentiated cells. It takes about 3 weeks or something for the cells to start myelinating, which of course is important to know in terms of time windows for clinical treatment. Now most of the current research efforts of my group is directed towards Mri Reporter Genes, which is the holy grail, I think, for imaging these cells. We use ferritin, we have used other iron oxide proteins that the cells makes its own iron oxide, and also special peptides that can be detected by advanced imaging techniques. And I'm happy to give some reference or papers from the last few years where you can look at that. We make artificial genes that have a lot of lysine-rich residues, and we can detect those amide proteins, which is interesting because we use naturally biodegradable peptides and sugars that don't contain any metal. We have even used glucose now as a cancer diagnostic instead of 18-FDG. So that's where a lot of development is, but given the time, and it's a very complex imaging technique, I'm not planning to explain this right now. So this is the group in 6th floor of the Miller Research Building at Johns Hopkins, about 20 people we have working on cellular engineering. Thank you. No questions, huh?
Well, sort of a clarification. Either I didn't hear this right, or I'm misunderstanding it. One thing you said was the standard immunosuppressants often don't work well in wellness.
In what then?
In wellness?
Rodents.
No, in rodents.
Oh, rodents.
Mice or rats, yeah.
Okay, thank you.
Like the standard cyclosporin, it's so bad for us if you're doing allogenic cell transplants. And when you do sort of cocktails, rapamycin, FKR5 or 6 cells survive for 60 days. With cyclosporin, they're gone at 1 week, and everybody is publishing cyclosporin, and they take the brain out after X amount of days, and they never have looked in the interim at the cell survival. So that's one of the important things. And we have also, and because of that, you also find other interesting things. Certain neural stem cells are also immunosuppressive. And in mice with EAE, which is a model for multiple sclerosis, we found out that these cells die after about 10 days when we put them in the ventricle, yet these mice show an improvement in disease that lasts for 30 or 40 days. So even when the cells die very quickly, there can still be long-lasting therapeutic effects. And one of the hypotheses is that they recruit endogenous cells. So even though they're only there a week, that during the time they can recruit enough endogenous cells to go inhibitory. So stem cells don't always need to survive.
I'm not sure if this is a question for you or for someone else, but is it okay to ask a stem cell question? Well, you can tell me if this is anyone's kind of this answer. I was interested in the issue of scars.
In what then?
I was interested in the issue of scarring.
Scarring. Yeah.
After a trauma. I met USA Today and we ran this powerful story this week about a poor Marine who had his face blown off by an IED. And he talked about how he's had something like 128 operations in 7 years, not just plastic surgery to reconstruct his face, but he keeps getting scar tissue over his eyes or over his nose that threaten his vision. And I was just thinking about the issue of stem cells and tissue engineering, and I don't know if people know something about that process of scarring, or is there research to understand how to control it, because that's regeneration, but it's not the kind you want. I don't know if that's a question.
All right, so the first question is, what causes scarring? It's like fibrosis or collagen formation, I think.
Would you mind using the mic?
The microphone.
Can't hear.
Oh, sorry. Yeah, so scarring is, I mean, I'm not a plastic surgeon, but I think it's primarily fibrosis or collagen formation scarring, right? So I guess that's a result of an inflammation or a foreign body reaction that is not suppressed, that get the collagen. But the use of stem cells, what I have seen is when you Google stem cell therapy on Google and you hit images, you get all these creams, to rejuvenate and this and that, and it's a stem cell therapy. So I think they can secrete a lot of factors maybe to heal that better. I don't know the molecular mechanisms, or to suppress the collagen formation. I know mesenchymal stem cells, they can increase angiogenesis. They can decrease collagen formation, often as related to immunosuppression. And they're known as the wound healers. These mesenchymal cells are actually used in burn wounds and repairs. So I think, I don't know, but I'm sure there's a whole body of research for mesenchymal stem cells and scar reduction, I'm pretty sure.
There's a big research on it, and this, some of what you're talking about is keloid formation, which is overgrowth of scars. That tends to be familial. It's also not distributed evenly over ethnic groups, and there are some things that can be done at the time of surgery to suppress keloid formation, but not to eliminate it. I don't work on that, but that's the sum total of my knowledge. It's partly personal. From an old wound I had sledding when I was about 10. It's like keloid. Kind of funny looking knee. But yeah, it's an awful thing for a person to go through, that kind of overgrowth.
So it's a different process than
It's abnormal.
the tissue regeneration.
It's related, but it's normally inhibited by contact. And there's a failure of contact inhibition when there's overgrowth, and that's a very complicated process. We know a lot about contact inhibition and kinases and intracellular signals involved in contact inhibition, but whether or not that had to do with this unfortunate serviceman's problem, I couldn't tell. So I had one question for you.
Yeah.
So these mesenchymal stem cells, when they're injected and appear to suppress inflammation, okay, do you know whether those are mesenchymal stem cells themselves, or whether that's a property of a cell into which they've differentiated? Because they can go in all bunch of directions. They can become osteoclastoid cells, adipocytes, fibroblastoid cells, etc. So do you know anything about that?
Right, so in the examples of these cells localizing in the brain with these lesions, in MS patients, it's possible they're starting to form bone. I mean, hypothetically it's possible, although it's unlikely. And the only way to image that is if we put those downstream lineages, these promoters that turn on, if we put a reporter gene in there, so then they will start lighting up if they differentiate into certain lineages, and if they don't, then we assume it remains an undifferentiated stem cells.
Well, if they go to osteoblastoid cells, it doesn't necessarily mean they're making bone. This is just stromal cell types. So,
Yeah.
so they can differentiate into several stromal cell types,
Right.
and they're distinguishable.
But not non-invasively, we have to take out the tissue.
No, what I'm saying is non-invasively, if you had reporters that were under control of Osterix, for example, or
Right.
any other, then you could distinguish between
Right,
osteroids.
right.
You described 4 different ways of putting mesenchymal stem cells into the brain. Are you at a point where you can draw a conclusion as to which of those 4, or maybe which 2 of those 4 are effective in terms of addressing multiple sclerosis, or is it still too soon to draw those kind of conclusions?
Right, so it depends on the disease, right? So Parkinson is clearly intraparenchymal, we know where we need to inject. Multiple sclerosis, we have multiple lesions, mostly around the ventricle. So the ventricular way, or intrathecally, is a very good route. Now what we're seeing is, and this was not really per se expected when cells are injected intravenously, there still are a lot of cells that end up there that do some beneficial effect. So the way would be to compare the IV versus the intraventricular administration, and we don't know yet what is best. I think the clinical trials with MS patients, MSCs, maybe had only started maybe two years ago or something like that, and the long-term outcome still needs to be all evaluated, so we don't really know. But it's important to know if your cells get there, because if they don't, the cells may still work, but they just don't get there, so you need to have other delivery methods. If you know that the cells get there, but there's no benefit, then the cells are no good, and you maybe have to use other cells. So you need to be able to answer the question, do they get them non-invasive?
Another question, you said that you consider MRI the holy grail as the way of marking and following.
Yeah.
Why is that the case? Why wouldn't, say, a good gamma-producing, radiation-producing marker
Okay.
be as effective?
Because we don't have an anatomical background, right? It's a hot spot, so we need to fuse it with an MRI is anatomical. You can see the tissue. With gamma camera or PET, you only see the tracers, so you still need to have the anatomy. It's done in combination with CT sometimes, but there's issues with overlay. Now, MRI is the highest resolution. It also can do these interventional injections. And optical imaging, such as bioluminescent imaging, is not possible clinically because the light cannot penetrate our whole body. So MRI, as I think, would be the holy grail. But the challenge of a very good reporter gene, for instance, such as green fluorescent protein, that something robust, that is a major challenge to do that, and that would be really advancing the
I
field.
mean, the MRI
MRI reporter gene, that's
Yeah.
the holy grail of all this imaging research, I think. But it's not there yet.